So welcome everybody to this webinar on Ozempic, semaglutide science, and using the Bio-Rad ELISA kit in order to kind of go after these concepts in your classroom. So this is a great contextual way to teach ELISA that could land itself well in AP biology, talking about basically signaling at a distance in A&P, because it's all about basically glucose regulation and hormonal signaling in order to take care of that, and in a lot of other kind of like physiology kind of related subjects. So like Lee said, we've kind of got our whole house here today as far as curriculum and training specialists. So hold on here one second. Now that I've stopped the annotation, let me move. Come on. All right. So Tamika is here. Lee Brown is here. The only person we're missing is Aaron Callis. Otherwise, you'd have every single person that does basically education support in the field for Bio-Rad kind of present today. Generally, our overview of what we're going to cover today is we're going to look at background information kind of on the history of semaglutide coming to market, and then how to use that to support students and ultimately constructing an ELISA to go about testing in the same way that semaglutide was first tested to figure out, does this work as a therapy for controlling diabetic A1C levels in the blood. And then we'll kind of show some of the other kind of resources we have around the ELISA towards the end. As I'm going through, if questions come up, feel free to throw them into the chat, and then Tamika and Lee can kind of break in, stop me, and then we can talk about those questions as you need to. So first of all, let me talk a little bit about Bio-Rad's ELISA kit, since that's what's going to be the hands-on thing that you would be able to use within this context. So Bio-Rad has a lot of resources related to their kits that you can get easily access to by following this QR here. This gets you to kind of all of Bio-Rad's resources for all of their kits. And that includes things like learning more about our biotechnology textbook. You can actually request a digital copy there to kind of take a peek at. It's got our webinars there, our upcoming workshops and things like that. You can also request posters and catalogs from there. And so this is actually a nice resource if you don't have stuff on your walls yet related to biotech. Bio-Rad has some really wonderful posters you can get for free by just going through this QR code, finding the posters link, and then adding the ones you want, and then requesting them. We also have linked up in there our biotechnology technique videos. So for any of the techniques that you might encounter in biotech, Bio-Rad's gone through and made short little videos that help students understand how to execute those techniques. They're really, really great kind of pre-lab pieces for any time you do anything remotely biotech. And that's everything from like using a transfer pipette all the way up to doing things like a Western blot and everything in between. And then also we've got some information there on the HOSA biotechnology competitive event that we basically helped HOSA bring to students a couple years back, and all of our support resources around that. The ELISA kit is going to be the core lab that would be used for the context we're going to talk about today. And this is actually a wonderful lab kit in that you get a lot of resources in one box. If you're doing the traditional way that the kit was kind of designed, which is to do it with like the spread of a disease in the classroom and have students figure out whether or not they've been infected by the disease, this would serve 48 individual students. Each student would have their own kind of body fluid that they would be monitoring to see if they got infected or not. There's actually 3 different scenarios in that original ELISA kit. And within that, we're going to take one of those out, basically the kind of quantitative ELISA, and we're going to use that within the context of understanding semaglutide and kind of glucose regulation today. And of course, with all the Bio-Rad kits, you have these manuals that you can access. They're free online. If you just go to the product webpage, you can download them and you can dig in very, very deep. Our manuals, to some people, are a lot of content, and that's why Control F is going to be your biggest tool with getting through them. So that way you can find just the background information you might want to know. You can find the protocol that is for you to set up the lab, for the students to run with these quick guides, and then of course the appendix that can kind of give you even deeper information on anything that might have been covered within the manuals. And like I said, one of the things that Bio-Rad does really well with these manuals is they've got these kind of quick guides that allow you to have something you could print off and give to students, or have a PDF that goes onto their digital readers for them to execute labs from. And the beauty there is they're really kind of clear, clean, and concise. It helps students kind of understand what it is they need to do, then has visuals to help support them in knowing what step they're working on. Within the ELISA kind of portfolio, we also have paper models which can be used to help students understand the theory how ELISAs work. ELISAs, especially for the first time students encounter them, are kind of a complex protocol, right? Especially if you're doing a non-direct ELISA, because then you're looking at potentially, this execution you're doing that's 8 to 9 steps, and that can be a lot for somebody to hold on to at once. And so I find that a lot of times going through and doing a paper model activity with students beforehand, before we actually do the in-person and hands-on lab, really gets them to the point where they understand the theory. So that way, when they're doing the different protocol steps, they've got a mental image of what's going on at that kind of molecule level as they go through and do it. Also on our website, we've got a general introduction for you in order to understand ELISAs in a little more detail if it's your first time doing an ELISA. And we'll kind of circle back to what an ELISA is after we do the background on more or less how semaglutide even came to market. And then finally, the other resources to be aware of, because the ELISA is a core technique that we use in our textbook. Bio-Rad has a textbook that can help you bring online basically anything from a couple week-long course up to a multi-year academy. This was written by Kirk Brown, who was an educator in the Central Valley of California, who had developed kind of a multi-year academy around taking students that had very little training in biotechnology, getting them hands-on skills and getting them prepared for the workforce. So the real focus here is kind of almost like a CTE edge to this, which is students walk away having skills that they can put on a resume and apply to jobs with. And so this is a really, really great resource, even if you're not teaching biotech yet, just to be able to kind of pull out theory from the book and some of the lab activities from the book, and then bring them into existing biology courses. And like I said, if you're interested in one of those, let us know and we can get you a digital copy, really easy to preview. And if you need a hard copy, you could talk to the CTS in your region, me, Lee, or Aaron, and we can figure out how to get you a copy. So without further ado, let's jump into the main kind of content for the day. So we're going to talk about the background information on semaglutide, Ozempic, and how it kind of gets its way to market. And so I jokingly with students would call this basically the history of dogs, monsters, and drugs. And that's because all of these things will basically play a role in this kind of blockbuster drug that we see in the market today. So semaglutide, right? Most of your students have heard of it because, boy, it just shows up in every single media stream out there. So it's kind of commercially ubiquitous. Everybody's at least heard of it. And we've heard of it in different ways usually. So we've heard of it under the different drug names where it's being used. And so the most famous one right now is Ozempic, Wegovy, RO, or other ones that are out in the marketplace being used for different types of therapies. And it gets a lot of attention because it has a kind of trizone effect. It's really good at controlling glucose levels. It also has some benefits when it comes to cardiovascular health. And then, of course, the one that has caused it to really, I feel like, end up in the limelight is its potential for weight loss control. But in the history of this, its initial goal, this original drug, was really brought on to treat type 2 diabetes. And so that's kind of the dig that we'll do is into that history and how that kind of comes to be. So why is even treating type 2 diabetes a big concept? Well, if you have been under a rock, you might not know that in the United States and kind of globally, we're seeing a great rise in the number of diabetics. And so in the US specifically, since the early 60s to now, we basically went from having like 1% of the population with diabetes, or at least diagnosed with diabetes, up to almost 1 in 4 people having diabetes. And this is an enormous growth. And this can be attributed to potentially lots of things, from changes in diet to a whole bunch of other things. And within this, we kind of have two different, types of diabetes to look at. And we'll dig into that in just a little bit. This ends up being an extremely huge market because diabetes brings on a host of other diseases, which end up having a large cost when it comes to human health and just overall cost to human healthcare industry. It's projected to be about a $413 billion total cost on the American market when you talk about the cost of diabetic therapies. And then that spirals up even more because it gives you, having diabetes gives you a risk for a whole bunch of other disease types, including blindness, kidney failure, heart disease, strokes, so cardiovascular damage, and then and really prolonged diabetes, like the loss of like toes and other extremities. So we have kind of two common types of diabetes that are recognized, type 1 and type 2. Predominantly, we're going to be talking about type 2 in this history. Type 1, we have about 18,000 youth are diagnosed with that every year. We have almost 6,000 youth that are diagnosed with type 2 diabetes every year. Type 1, there's not really a great way to prevent this. It's one of these things that the body just doesn't make enough insulin. So you've got kind of damage sometimes of where insulin is being produced in the pancreas. And type 2 is one of these ones we feel like we can get control of. Most cases seem like they could be prevented. And this is really where you see a lot of the medications coming into the market, because if you can regulate how insulin is being moved about in the body, you can decrease somebody's chance of becoming a full-blown diabetic. So in general, when we talk about diabetes, right, we're talking about a chronic disease where the body doesn't produce enough insulin or can't use insulin in order to regulate glucose levels in the body. So type 1 is that autoimmune condition. And so that's why this is a little bit harder to treat than the type 2s. We have insulin producing cells that are attacked or other things are going on, they just don't produce insulin. Where type 2 diabetes, you've got a kind of like a hormonal condition. You've got just basically less insulin being produced, or the body doesn't know how to respond to the insulin. Like it doesn't get a high enough spike of it in order to make decisions about pulling glucose out of the bloodstream. And diabetes is something that's been around for quite a long time. So, I mean, even historically, one of the first sort of physicians that we were aware of, Hessia Ra, who's actually technically a dentist, but you have to remember in olden times, physicians and dentists were more or less of the same cloak. You have Hessia Ra in Memphis in Egypt in almost 2700 BCE, had recognized this disease state. And that was basically that they could smell and notice that there was additional sugars in the urine. And then also talked about these people that were producing these sweet urines, that they would just lose body weight like crazy. So there wasn't really a treatment. It was more of just like, we can recognize that there's this disease state going on. And then if you move over to India in about 1000 BCE, the Ayurvedic physicians also were able to recognize people that had diabetes due to this kind of like honey urine, this also sweet urine aspect of it. And they went even further and were able to break down kind of diabetes into, they had kind of more or less 3 types of diabetes they recognized. One was kind of our type 1, one was kind of the type 2, and then one was this thing that was more or less a type 3, where the patient more or less always had these burning sensations. And so it's something that's been recognized for a very long time. Diabetes as a term really doesn't get coined until the Greek Apollonius of Memphis kind of gives it that name, and diabetes more or less means to, in the Greek, to flow through or to siphon. And coincidentally, Apollonius is living in Memphis, which is the same place that Hessia Ra was in Egypt. So when the Greeks are basically colonizing Egypt, Apollonius is in the same place as Hessia Ra, so I've got to believe that a lot of his knowledge actually is coming originally from Hessia Ra when it comes to diabetes. Our next big jump in kind of the history of diabetes really doesn't take place until almost 100 years ago. And this is where you start to get the first, I guess, chance to basically find ways to deal with diabetes at kind of a biochemistry level. So in Europe, in the 50 years kind of prior to this slide, there's a lot of Europeans going through and understanding what different organs are doing and what disease states they seem to be kind of associated with. So you have a German by the name of Langerhans who figures out that the pancreas is it seems to be very important in potentially the regulation of blood sugars. And he specifically is kind of a physiologist, and he finds different cell types inside of the pancreas. And that, to some degree, leads to Banting and Best in Toronto, who start to experiment with dogs to figure out if some of these things that are being produced from the pancreas can help regulate kind of glucose. And so they're going through doing surgeries on dogs, removing the pancreas, noticing that they can cause a diabetic state in the dog, and then attempting to take some of basically the factors coming out of pancreases and then adding them back to dogs to see if you can rescue a dog from the diabetic state. And so using this methodology, they ultimately discover insulin. And unlike contemporary days, you discover something and then you go into clinical trials for half a decade or more, and a couple millions of dollars go away, and then you eventually get to test it on people. Within basically 1 year of their discovery of insulin, they have the more or less first human trial, which is Thompson, a 14-year-old type 1 diabetic, who ends up getting injections of this factor that they're finding and calling insulin. And it seems to regulate his blood glucose levels just like it was doing with their dogs. Shortly thereafter, you have the discovery of glucagon. And so you can kind of think of this as the yin to the yang of insulin. And this glucose antagonist, as Kimball and Muirland kind of describe it, is also a pancreas extract. It's coming out of a separate set of cells than the ones that you find insulin coming out of in the pancreas. And these basically, instead of getting glucose to come out of the blood and into cells, does the opposite. It helps kind of glucose come out of storage and into the blood system. And shortly thereafter them, in 1925, you have the discovery of what was called these increase insulins in Belgium by Laber. And these are really interesting because these other two are being produced in the pancreas. And these incretins were more or less discovered because people were watching those glucose levels and then realized, you could regulate them with insulin and glucagon. But then also they realized that if instead of, like, say, injecting glucose into somebody, you gave it to them orally, what they noticed was all of a sudden you had this increased effect of insulin that was different than, say, if you injected insulin into the patient. And so what came out of that was this idea that there must be something coming from the gut that recognizes available glucose and more or less triggers the production of insulin. And so this ends up being kind of the tip of the iceberg to ultimately what will become these drugs like Ozempic. And so these increased insulins kind of get condensed into the word incretins. So if we dig a little bit deeper into insulin and glucagon, we can kind of talk about what we know and what we don't know about them, because we sure do know a lot about them, but there's still other things they're probably doing that we don't have a firm grasp on. Because remember, these are acting as hormones. They're going into our systems and causing lots of changes all over the place. So glucagon increases glucose in the blood. It's produced in the pancreas. It's coming from the alpha cells in the islets of Langerhans. It's a peptide hormone. It's relatively small. It's only got 29 amino acids, and it's derived from proglucagon. And this is only important because proglucagon ends up being this thing that we find in different tissues, and its regulation and transformation ends up giving us some of these other hormones that become important in the regulation of glucose levels. And it has a really interesting regulation by glucose. So if you've got low glucose in the blood, what will happen is you've got less energy basically getting into cells, right? So if you're in the islets of Langerhans and these alpha cells, what happens is your ATP level in those cells start to drop, which means you'll basically kick on the potassium pumps and or your potassium pumps don't work as well. So potassium starts going up in the cell, and you'll build up so much potassium in the cell that you kind of get almost like almost a depolarization event as calcium tries to flood in, which causes excitosis of glycogen. And so that gets dumped out, which then will cause a spike in glucose and then kind of get this thing back into a general regulation. However, glucose is the main thing people focus with glucagon on as far as being a regulator of it, but you have a lot of other things that have feed-ins to this pathway. So that's everything from insulin to somatostatin to zinc, amylin, GLP-1, and GIP, these incretins. And it has a lot of functions, and some of these are still being figured out. When we get to insulin, it's doing just the opposite of glucagon, right? It decreases glucose in the blood. It's also produced in the pancreas, but instead of the alpha cells, it's in the beta cells. It's also a peptide hormone, but it's a little bit bigger than glucagon. It's a quaternary structure with an A and B chain that come together, and it's stored basically as a hexamer around a zinc atom in these cells. It's regulated by glucose, and that basically recruits transcription factors and coactivators to a promoter that then kind of help drive its production. But it has a lot of other regulators that come in and modify how much is being released. And that's everything from melatonin to estrogen to leptin to human growth hormone, GLP-1, GIP, adrenaline, and noradrenaline. And just like the other one, right? Any sort of hormone, because it can go out and touch all these different tissues, has a lot of functions. We know a lot of them, but there are still other ones I'm sure that we will find as we continue to play with these systems. Looks like we've got a question there. Norman?
Yeah, hey Damon.
Yeah.
So I've heard that insulin production is really upregulated when sugar floods into the bloodstream. Like if you drink like a high fructose syrup soda, that can really spike your insulin production
Yep.
in your blood.
And that's kind of happening for two reasons if you're drinking it, right? And that's because you'll ultimately get the glucose that's going to be circulating because you're getting a bunch of like sugars that are breaking down really quick in the bloodstream. But even before that, because it's going through your GI, as that sucrose is being broken down in the GI tract, will actually start triggering the things on this slide, which are going to be these incretins, things like GAP and GLP-1, which can also upregulate the production of insulin. And so there's kind of two pathways that end up working when you ingest something with sugars in it. And so using that kind of as a jump off for this point, the next kind of portion of the biochemistry era happens in like the 1970s. And this is when that stuff that Labar had seen, which was if you give a patient the same amount of glucose orally versus injecting it, you get a bigger insulin response. That comes down to these two hormones that are being produced in the gut. And so the first one that's found is the gastric inhibitory polypeptide. And it's originally found to kind of stall out the gut with gastric acid release and increase in insulin. And then 1981, people start realizing that they can find proglucagon being expressed in the intestines. And that's not something that, made sense at first. But it's not glucagon yet. So you've got some tissue specific kind of differential processing that happens in the gut to take this proglucagon and turn it into GIP and GLP-1. In 1983 is when we actually find GLP-1, which is ultimately what's going to become semaglutide in the long run here. And it's a small peptide that's derived from that procoglucon in combination with GIP causes this incretin effect. And that incretin effect, again, is when you have basically glucose hit the gut, you get this hormone response of these being released that then upregulate insulin in the blood. And so the way to think about this is the body is basically getting ready for a flood of glucose because a meal is coming down the pipe. And so it's kind of pre-gaming the system for this glucose that's about to hit the bloodstream. And so that's part of this kind of like what I'd say is like a multi-level regulation that's going on when it comes to our blood sugars. And so just to kind of put both of them in context again, we got GLP-1 and GIP. GLP-1 will decrease basically the desire for food intake. And that's more or less because it's telling the body, hey, we've got enough glucose coming in because I'm having to turn on insulin to kind of bring it down. And this ultimately ends up bringing down blood glucose by turning on the secretion of insulin. It also slows down kind of gut mobility. And then GIP, on the other hand, also brings down glucose, increases in insulin secretion, but doesn't seem to have as many targets in other tissues as GLP-1. And GLP-1 definitely seems to have the largest effect between these two incretins. Both these are polypeptide hormones. They're released in the GI tract, And specifically, they're released in K cells in the duodenum and jejunum. And with GLP-1, it targets the pancreas and it targets the brainstem, or at least these are two to think about. It targets some other places, but these are two really important ones. It targets pancreas, right? Because that's where you're going to get your insulin production from. But the brainstem is really interesting. And this is going to be a factor when we ultimately talk about some of the side effects of these drugs. So GLP-1 will hit the brainstem in the nucleus of the solitary tract, area prostrama, and it ultimately will cause nausea at certain levels. And this is why when people take semaglutide and some of these other drugs, they end up feeling nausea. And nausea, right, is a great way to get something, an organism, to stop eating. And this is probably why if you just get a high enough titer of GLP-1, you feel nausea, because it's ultimately a feedback function to decrease the desire for food intake. If people were trying to use these drugs, especially like say GLP-1, because it has that impact on desire for food intake, and in addition to regulating kind of blood glucose, the only bummer is if you're trying to use this just straight up as a drug to give to patients, is it has got a really, really short half-time. It's got a half-life of 2 minutes. So it's a really quick, regulation system here. And it's actually, the way that this works is when it gets released in the bloodstream, it's quickly cleaved by this protease DPP4 that can target this compound and then break it down really, really quickly. So this makes GLP-1 as it is not a very good potential drug to say treat diabetes with, or to even say treat people to curb their appetite and things like that. But coming out of this era, what we get is we get a much larger picture of basically regulation of glucose in the body. So instead of just the old story of insulin and glucagon, we now have these incretins involved, GIP and GLP-1, which create these huge feedback loop systems, right? Where you can, before glucose even hits the bloodstream, your body can be like, I'm going to get hit by a bunch of glucose. I'm going to increase my insulin in order so that way I can kind of store it. And then once the glucose is out there, depending upon the level of it, it can basically trick on the next level, which will kick on another level of insulin production in the bloodstream. And so you get this really kind of complex feedback pathway that ultimately gives you homeostasis, right? And that's the whole goal of all of these hormonal systems, is to give you homeostasis, where you've got a level of glucose that allows you to have energy but doesn't cause damage to organs. So if we talk about how could we turn one of these things actually into a drug that might help people that have difficulty regulating their insulin levels, i.e. type 2 diabetics, we really don't have a breakthrough until the early 2000s. And it kind of comes out of left field. And this is where the monsters part of the history comes in. The breakthrough is not physiologists or people really in the medical industry studying this. It comes from basically herpetology nerds that are really into understanding how venoms work. And so people are studying the Gila monster, which is this lizard you can find in the American Southwest. And it's a really unique lizard in that not many lizards produce venoms. There's just, I think, one or two of them in the world, maybe, or maybe a handful more. And so people are trying to understand what is in the venom, because people have been studying snake venom for a long time, and we've gotten a lot of tools out of snake venom. So folks are trying to figure out what are all these components that we find in the venom of the Gila monster. And in the process, this compound gets found, or this peptide gets found, that is called exenidin, or exenidin 4, or exenidintide. And it was originally a similar compound was found in the beaded lizard in Mexico, which is similar to Gila monster. It's also a venom producing lizard that looks pretty darn close to the Gila monster. But the interesting thing about this peptide in the venom is that it's basically GLP-1, but slightly modified. And the modification makes it really, really special to someone trying to regulate glucose. And that is because the modification that takes place between GLP-1 and exenidin 4, this thing in the venom, is it changes one of the amino acids where DPP-4 cleaves. So all of a sudden, that protease can't break down GLP-1 as fast. So instead of having a 2-minute half-life in a bloodstream, it's got a half hour, or sorry, it's got a 2-hour half-life. So that's huge. All of a sudden, that is something you could work with as a pharmacologist. And so this basically goes straight into trials and comes out under Amylin and Eli Lilly. And it's basically a synthetic form of this exenedin 4 that they're finding in the Gila monster. And they find that it survives much longer in the human blood system. It's a great candidate for being used for type 2 diabetics, but it would be way, way better if you could even give it a longer half-life, because with a 2-hour half-life, you kind of have to dose somebody really, really frequently to get this to be very useful. So in 2010, and as soon as this comes on the market, every company is looking at this and going, there's big potential in here. We got to get our biochemistry, jockeys in here to go through and start tweaking with these amino acid structures to see if we can get this thing to last longer. So by 2010, Novo Nordisk releases this thing called liraglutide. And basically what they've done is they've added a side chain of a fatty acid onto this, which basically helps protect it a little bit in the bloodstream. And they've done a couple other substitutions to make it so it doesn't get cleaved as fast. And now you've got this thing that's 13 hours long. So that's pretty good. You went from 4x to another almost 4x, or more than 4x of the lifespan of this thing. And then ultimately in 2007, you get semaglutide that comes out. And so this keeps that DDPP4 protection. It adds a longer fatty acid side chain to it and does a couple other substitutions. And all of a sudden, we've gone from this thing that has a 2-minute half-life to a 7-day half-life. This now is something you could see somebody taking and being able to regulate their glucose without basically having to be there every hour, every 2 hours with an injection. And so this is basically the big kind of breakthrough, or these changes to this venom that we got from the Gila monster. So just to kind of give you a visual of that, here's GLP-1 on the left, and here's semaglutide on the right. And so the big change, right, that we find in the Gila monster is this change in the amino acid here, which gives you protection against DDPP4. And then the other big thing that comes along and is the big modification is we've added a big spacer region onto the edge of the amino acid with a really long C18 tail to it. And this basically just increases kind of this fatty acidness of the peptide. And what does that do? It means that it can easily bind to albumin in the blood, which will ultimately protect it from other proteases coming in and trying to kind of cleave it. You can kind of cover yourself in albumin to some degree. And then the final thing is they do this lysine to arginine substitution at position 34, and this helps drive your ability to basically add that really long fatty acid chain on there. And so this ultimately is what gives us this blockbuster drug, is finding it in the venom of the Gila monster, doing some modifications to make it even a longer half-life. And now we've got this thing that is, from a pharmacist's point of view, something you could give to a patient and do it in a way that they don't have to be there all the time. But if you think about this in a big biology context, one thing that people always ask is, well, why the heck is this even in the Gila monster? Like, what benefit would this be in Gila monster venom? So I want to just do a little side note of what's going on there. So Gila monster venom is really complex in that you've got a lot of active compounds in there. You have that GLP-1 antagonist. And what that's doing is, imagine if you're a Gila monster, you're a small, not small, but like mid-sized lizard. You don't run very fast. You bite a prey, which tends to be things like rabbits and squirrels, and your best bet is to kind of hold on to them, but you always can't do that. They do kind of have like pit bull like jaws that can hold on to stuff, but sometimes the thing runs off. But if you can get your venom in them and it's got that GLP-1 agonist, what's that going to do? It's going to increase insulin production, right? Which is going to do what? It's going to pull all the glucose out of their bloodstream. What happens when you've got no glucose circulating? You become lethargic. You can't move very fast, and so you're not going to be able to run away from this lizard. The other things in the venom is a giant dose of serotonin, which basically just overrides the nervous system. It causes pain, probably in the animal. It causes blood vessel constriction and then muscle rigidity. So once again, it's keeping that animal from getting away. It then also has this giant cocktail of proteases, and a very specific one in there is this gilotoxin, which is basically a protease that goes after neuronal tissue. So besides causing the pain in the neurons, you're also going through and actually starting to break down neuronal tissue. So now you can't get functional, basically muscles and things like that. Then the last thing that's in there, as far as a component, is hyaluronidase, which destroys connective tissue. And so more or less the organism starts breaking down before the Gila monster even starts eating it. And so for Gila monster, this works because they bite the organism, they inject the venom using their teeth, and that's what you can see on the left there is those sharp teeth, probably the scariest teeth of a lizard I've ever seen. And then it basically shuts down the organism from being able to run away. And so the Gila monster can kind of follow them until they more or less die and then go through and eat them. So the semaglutide, now that we've got this modified protein hormone that we found in a lizard, has all of these targets when we look at it going into a human body. The main ones that people are focused in on from like a therapy point of view, of course, is the targets on things like the pancreas and like the brain, because it's going to help us regulate glucose, and it's also going to ultimately decrease the desire for food. But it has all of these other tissues that it's hitting. And so from the pharmaceutical industry, this is really exciting because as you maybe tweak this molecule, you might be able to get other effects from it that can work on other disease states. So the main uses, though, that people focus in on that went through clinical trials first was regulating kind of hyperglycemia, right, for type 2 diabetics by changing insulin levels. And then I think some of the downstream ones we'll see are people going through maybe tweaking it to get different sort of cardiac therapeutic effects from it. The only bad side, though, about the semaglutide and the similar compounds that are being made is a lot of these have nausea associated with them, which isn't too surprising if we remember GLP-1, right? GLP-1 is going and hitting the brainstem and causing that kind of nausea effect. And so regulating that is always something that's going to be difficult, I think, with these drugs. And since we're just at the starting point of using these, we also have to kind of keep our eyes open for other side effects that might tell us other maybe long-term effects are taking place. But that being said, they've been a blockbuster when it comes to type 2 diabetics because it allows them to really, really regulate their glucose levels. And then kind of coming out of that, we've seen as an kind of an accident from the first couple studies that were done trying to do the type 2 diabetes that it's really, really good at regulating weight loss. And this is where it's really kind of moved into the popular kind of imagination. So if you were trying to study this drug when it first came out, and you're trying to look for its effects on regulating glucose, you could kind of do this in two proximal ways. You could look at the regulation of insulin, but a more direct way would be to actually look at the regulation of glucose levels in the blood. And so the way that you would normally do that is to be looking at glycated hemoglobin, or what would be known as HbA1c, or as it's always called, just kind of in general speak, A1c. And this is basically the amount of glycation that you find on hemoglobin floating around the blood. So normal people have usually less than about 5.7% glycated hemoglobin floating around. Pre-diabetics have about 5.7 to 6.4, and then diabetics are those people that we find highly glycated hemoglobin over 6.5%. So some of the first studies, if you go and dig into the literature, you can find are when they're taking basically semaglutide and then following patients and seeing how it affects their A1C over the course of time. And so ultimately what we see is when people start doing trials on this is that if you take, a cohort of people and you that start off as kind of like diabetic level or pre-diabetic level, and you start giving them semaglutide, that over the course of time, those that are on semaglutide start to bring their A1C levels down. And this is ultimately what you see in a lot of the clinical trial data from semaglutide. Also, on the side, people noticed when they were doing these studies that they also saw kind of body weight come down. And this is ultimately what leads to kind of Ozempic as a drug. So as an educator, what you can do is you can set students up for kind of investigating what these original trials looked like for semaglutide in terms of regulating A1C. And the ELISA is going to be a great way to basically put a metric on whether or not the drug is working. So the idea here is that you can kind of do a mimic study with students. And so what you need to do is understand your students, understand what level of this background knowledge they need in order to understand how semaglutide is working to regulate glucose levels, and then have them, or kind of Socratically lead them into designing an experiment by which they'll do a very similar kind of experiment to what we just covered, which is taking some folks that are diabetic or pre-diabetic, giving them semaglutide, and then seeing, do we see a change in A1C level over time compared to a control group? And so for their knowledge, basically, this is what they would have to understand is kind of that feedback loop that we've gone through a couple times of how insulin, glucagon regulate glucose in the bloodstream and how GLP-1 semaglutide fit into that picture. And if you need something to kind of give them a great background on how A1C works, there are some great YouTube videos out there. So this is one that you could use as kind of part of the pre-lab, so you understand how A1C is well regulated and also diagnosed. And then you could kind of lead them into the study. And so there's different ways you could assess the A1C. You could use HPLC. None of us really have access to that usually in a classroom. Enzymatic tests or ELISAs. And ELISAs tend to be what a lot of people will use because they're really easy to work with in kind of like a non-high-end lab space. And that's why you see ELISAs used to diagnose things as like diseases, et cetera, in all kinds of places. And so for the students, some of the things they'd want to consider for setting up their study is, are they going to give semaglutide orally? Are they going to give it subcutaneously? Oral semaglutide does not have a great kind of percent hit as far as driving what you would hope. And so you would kind of want to kind of convince students that they'll probably want to do this injectable semaglutide delivery. And then also, like, how are they going to monitor this? And so ultimately, they're going to do this through an ELISA. So with Bio-Rad's YouTube video playlist, we have how to do a quantitative ELISA on our playlist. And this is a great place to start if you've never done a quantitative ELISA before. It's also a great pre-lab to have students look at before they attempt to sort of design their mimic study. So ultimately, what I would kind of drive students to is that we need more or less, two groups of patients. We need a control and we need a test group that will be giving semaglutide to. We kind of use Socratic method here to kind of get them to the point where they're like, well, we want to use the injectable just because it has a bigger effect, as far as we can tell, on getting to the cells and causing potential regulation. And so within the study, you would have them kind of do like weekly injections and then monitor these patients every week for their A1C levels. And the way we could monitor them, like I said before, is we would use an ELISA just because it is an easy to access tool compared to HPLC or some of these other enzymatic tests. And so for them, in order to use an ELISA, they kind of have to understand how an ELISA is constructed. So an ELISA, if you haven't been exposed to it before, stands for an enzyme-linked immunosorbent assay. And in the elevator pitch, it's basically using antibodies to detect specific compounds. And most of the time, it's going to be proteins. And then using an enzyme linked to the antibody in order to drive a color change that allows you to tell, was my original protein or item of interest there or not? And there's a whole bunch of different ways that ELISAs can be constructed. And so you could have a direct ELISA, which basically just takes an individual antibody, has an enzyme linked to it, and tries to detect that specific antigen and then give you a color change. The only problem with these, though, is they're very, very slow, because you usually have a hard time getting a high titer of the antibody in there. So most of the stuff that you run across when you talk about ELISAs, if you go into a clinic and say, get a strep throat test and stuff like that, are these indirect or sandwich ELISAs. And the reason that these are so powerful is that they will basically use two antibodies in order to give you signal amplification. So for everything that you're trying to detect, every antigen, it will be able to bring multiple antibodies in with the enzyme attached to them, which basically increases the number of enzymes there, which will then make the rate of reaction much quicker. And so that way you can go in and you can get results within 10 minutes instead of being there for hours waiting for this slow enzymatic reaction to take place due to concentration. So what we're going to use for this mimic study would be a sandwich ELISA. And so what that's going to do is it's going to take an antibody that's going to be bound to the plate. It's going to capture your target molecule. And in this case, it's going to be that A1C. And then it's going to have that be probed by a primary antibody that can recognize that. And that primary antibody will be able to have multiple secondary antibodies that carry the enzyme attached to that. So that gives you the signal amplification. And then when we add the substrate there, you can then get a really quick color reaction that tells you whether or not that original antigen, A1C in this case, was going to get trapped. So like I said, ELISAs get used for all sorts of testing: disease diagnosis, hormone detection, drug detection, GMO detection, cancer biomarker detection, allergies, and research, like what your students are going to do with this. So typically, this would be done in a large microtiter plate, but what your students are going to do is they're going to work with individual microtiter strip plates. And this allows kind of each group of students to run everything they need just in one easy to handle strip. And the way that our ELISA is constructed, you can do this qualitatively with them, so they can just compare the color change against a set of standards that they are actually going to build by doing a serial dilution. Or you can do this quantitatively. You could use a spectrophotometer to diagnose exactly how much of the product is there. And that will tell you, you'll be able to do a correlative linear regression to understand exactly how much of your target was there. So ultimately, I'm going to run you guys through the protocol kind of quick, just to give you an idea of what this looks like for the students. At each station, what they would have is they have a microtiter strip plate. And what they're going to do is in half the plate, they're going to make a serial dilution of our target. And then in the last 6 wells of the plate, what they'll do is they'll have a patient from the control study and a patient from the treatment study that's getting the semaglutide. And so students would basically go through, they would label their plate up, and what they would then do is add their samples of A1C to the first couple wells and then do a serial dilution. And so they'll basically start off with kind of like 1,000 nanograms per milliliter in the first well and just perform a serial dilution so that it basically drops in half every time. And then in the final well, you just wouldn't add anything besides the buffering solution PBS. And so that way you've got a complete zero there. And then in these other 3 wells, that's where they will add the patient samples that they'll be kind of analyzing to see, are we seeing a regulation in A1C in these patients over time? And so when they add the materials in there, what's going on is you'll have antibody that's stuck to the bottom that's basically going to capture the A1C on the hemoglobin. And then what students will do is they will wash those wells, and that'll basically remove anything that was in the samples that wasn't the target. So now all that we'll have is the antibody with the antigen of interest, the A1C in this case, stuck to that, and then everything else gets washed away. When you do this wash step, the other thing that's taking place is it's also coating the rest of the wells so that no new proteins can randomly stick there, because we're going to ultimately be probing this with antibodies, right, that are large proteins, and we don't want anything else to randomly stick to the well because it might give us a false positive. And so that washing of the wells after the first addition, it does 2 things. It removes the leftover materials from the patient samples and from the controls, and it also coats the plates so that you don't end up with a false positive. The next thing students will do is they'll add their primary antibody that will go through and recognize the A1C that's been bound to the sandwich of antibody that's on the plate. They'll then wash that off after a short incubation. And what will happen after that is they will add their secondary antibody. The secondary antibody is going to bind now to the primary antibody that's stuck to the A1C, that's stuck to the antibody that's holding on to the plate. And this is what's going to bring the enzyme into each of the wells if the A1C was there, right? And so what will happen is, so I'm just going to skip through how to do that, is that now that when you add the substrate, what you'll get is you'll get a color change. And how much of a color change you get is going to correlate directly to how much A1C was present. And if students look at that now compared to their serial dilution that they did, they could just eyeball this and say, I've got about this much A1C concentration in my specific patient. And this will allow them to start to piece together as a group what's going on with semaglutide if you're giving it to patients over the course of 16 weeks. And ultimately what they'll see is, as they kind of pair this data together, is that in their placebo patients, the PPP patients, they won't really see an A1C change throughout all of these 16 weeks. Where with the treatment patients, the TP patients, what they will see is they'll see a gradual decrease that actually kind of takes place relatively soon in the first couple of weeks, and then kind of holds steady, that brings the patient kind of out of the diabetic level of A1C into the pre-diabetic zone. And so this ultimately allows them to see what those original tests with semaglutide were like, that then proved that this could be a wonderful medication for type 2 diabetics. If you've got a microplate reader, you could put all those strip tubes just back into a plate, snap that in, and then read it using 650 nanometers. If you're reading it well, the plate is still blue. Or in the back of Bio-Rad's manual, there is a way to stop the reactions with acid, and that'll change those wells from being blue to yellow. And that allows you to say, come back on another day and then read them because it's been paused out. And if you do that, you're going to read it with 450 nanometers. But ultimately what the goal here is for students to be able to notice the change in A1C that takes place when semaglutide is given to patients over the course of time. So ultimately their data set as a combined class ends up looking something like this. You know, those placebo-controlled patients, we really don't see much fluctuation in their A1C levels, where those that were getting semaglutide, we will see a drop that gets them hopefully out of that diabetic state. Since we're kind of running close to the end of time, I'm going to go ahead and pause there. Some of the rest of this is just kind of nitty gritty about how to set up the prep and things like that. And I'll handle any questions that may have occurred that I wasn't able to get to during the bulk of the presentation. Lee and Tamiko, is there anything that came up in the chat that I missed that I should get back to?